Hello, my name is Nan-ShanChang. I'm from Taiwan. Today I'm going to talk about normal cells repair WWOX negative or dysfunctional cancer cell via WWOX cell surface epitope 286 to 299. We have first cloned a gene called WWOX. This was in year 2000. So you should be able to see there are several semantic issues you may be interested in. In 2020, NIH announced that WWOX gene is one of the 5 newly discovered genes for Alzheimer's disease. WWOX has N-terminal double domain. There are 2 of them. The first double domain has 2 tryptophan and 1 tyrosine 33 phosphorylation site. And there's a C-terminal short-chain alcohol dehydrogenase domain. SDR domain has NSYK motif, which binds to estrogen or androgen. N-terminal double domain has conserved phosphorylation sites such as Y33 phosphorylation. And the gene is huge. It's about 1.1 million bases, 9 exons. So it's a candidate tumor suppressor. So under stimulated condition, WWOX undergo phosphorylation is tyrosine 33, and it goes to mitochondria or goes to nucleus to induce apoptosis, and this work together with the p53. But JAK1, c-Jun, and terminal kinase can block it. So when knockout mouse model was established, WWOX shown to be participate in bone homeostasis, osteosarcoma, and associated with HDL cholesterol and triglyceride level and type 2 diabetes, and also with aerobic metabolism, audiogenic seizure. WWOX knockout mouse has a reduced body size, loss of subcutaneous adipose tissue, and death of spacious gland cells, alterations in myelin sheaths, and movement disorders such as or ataxia. Epilepsy can only survive for 3 weeks. In human, can survive about 2 years or longer, a little bit longer. Rats 2 months. WWOX has so many binding proteins. In the W domain, it binds to PPXY or PPPY motif, such as in p53, AP2 gamma, or BB4. Or when tyrosine 33 is phosphorylated, it binding so many other proteins. And SDR domain in the C-terminal also binds quite a few proteins. So today we are going to focus on TGF-beta binds to HER2 and WWOX. TGF-beta binds to type 2 receptor and then recruit type 1. And type 1, type 2 receptor together signaling downstream SMAD2 and 3 and complex with SMAD4 to induce either gene expression or induce apoptosis. We have first discovered a novel TGF-beta signal pathway. So basically membrane hyaluronidase HER2 can act as a receptor for TGF-beta and hyaluronan. TGF beta can induce nuclear accumulation of WWOX. So for example, this L929 cell also is stimulated with TGF beta. You can see SMAD4 goes to the nucleus, and WWOX also goes to the nucleus. But HCT colon cancer cell, HCT116, if you stimulate with TGF beta, WWOX also goes to the nucleus. But these cells does not have TGF beta receptor. So we're looking into what receptor may be responsible for the effect of TGF beta, which shows that it binds to membrane HER2. In the immunoelectron microscopy, you can see a large particle is HER2, small particle is TGF beta 1. They colocalize together, suggesting that they bind to each other. And the complex HER2 and TGF beta complex, it can internalize as an endosome and then go complex with the lysosome, and then may be degraded. Actually, WWOX by itself can localize on cell membrane, says this starch, and the complex between WWOX and HER2 shown on the cell membrane. And when the cell is stimulated with TGF beta, it goes to the nucleus. And some of the complex still on the nuclear membrane. This is much clearer. You can see TGF beta induced more complex goes to the nucleus of the HER2 and WWOX. So in this pathway, TGF beta binds to HER2 and WWOX complex, and the complex relocated together with SMAD4 to the nucleus to induce cell death or cell proliferation. Cell surface hyaluronidase HER2 is a cognate receptor for TGF beta to activate tumor suppressor WWOX for regulating cell growth and death. So we are thinking about WWOX could be a sensor of extracellular cue, because WWOX is localized. A portion of WWOX is localized on the cell surface. Actually, is anchored by HER2. I'm going to show you how WWOX deficient cells meet WWOX positive cells. So there's a strategy must be developed by WWOX deficient cells. There's an inverse relationship between HER2 and WWOX. So if you knock down WWOX, HER2 level is significantly increased compared to scramble control. So if WWOX is overexpressed, HER2 expression is suppressed. And cancer invasion occurs if you silence WWOX. There's an increased migration. So you can see when WWOX is suppressed, there's certain amount of cells, number of cells goes to the lymphatics very fast, compared to this overexpressed WWOX, and the tumor gets suppressed. And here's the normal control. So in this cell migration assay, actually it's wound healing assay. Wild type MAP cells is sitting on both sides. Unfortunately, after 48 hours, the cell doesn't heal. In knockout cells migrate individually really fast. So you can see wound healing very well. Next, we on one side of the chamber, that's from EBD. One side of the chamber, we sit WWOX negative, and the other side we sit WWOX positive, like normal cells. And you can see in this video, you can see knockout cells, MAP cells, migrate individually, wild type migrate collectively, and knockout cell undergo retrograde migration. They used to be brother sister. They can hug each other, but no more. But without WWOX, they can no longer recognize each other. So this slide shows WWOX negative cell undergo retrograde migration upon facing WWOX positive cells. So we do a tracking. You can see R, that means retrograde migration. Actually, just see the video already. So how can we make both sides recognize each other, even from different species? Okay, even they not used to be brother sister. So we found that one of the antibody we can do it, and then migrate forward is antibody against MIF, migratory inhibitory factor. So this culture, you add antibody, you can see 231 migrate forward. A means enteral migration. In control, the cells they undergo retrograde migration. So in addition to MIF, we also shown that antibody against ephrin also abolish the retrograde migration of the WWOX negative cells. So all of them, ephrin A1, B1, B2 antibody. So this molecule directs the migration of cells. And so during migration of 231 versus L929, you can see wing pathway. Okay, this is a major real-time promoter activation. So wing pathway is get activated really fast in 231 cell, but not in L929. Also, NF-kappa B, it only occurs in L929. Creep promoter occurs in L929. And SRE is just for ERK, MIC pathway. And basically, not much differences. And you have inhibitor for the wing pathway XFB393, and this allow retrograde migration turn into the anterograde migration. So just like this. So this study suggests that wing pathway, yes, is involved in the detection of the retrograde migration. Next is TGA beta can do the same way. So if we add TGA beta in the culture, originally retrograde migration now become anterograde migration. So don't run away. They merge to each other. Okay, 231 is human. L929 is mouse. So basically, TGA beta can make two different species cells merge together. Here, all the signaling we just mentioned actually convert to ERK for retrograde migration. So here's the time course by time-lapse microscopy, and you use U0126. That's a MIC inhibitor. So downstream signaling for ERK and so on is blocked. And under this block, you can see both sides of cells, they merge each other. So suggesting that MYC, from upstream, RAS, MYC, and ERK. This pathway is important for cell-to-cell recognition. This slide showing that WWOX negative and positive cell, they meet each other. There's significantly increased oxidoreductase activity, redox activity. So you can see red color here, really fast. And as a consequence, the cell die in the wild type cells. So you can see the DNA fermentation. So a typical apoptosis caused by WWOX negative cells, or we can call it dysfunctional cell. So if we restore WWOX in WOX negative cells in one side by 5-ASA and see what happened. So WWOX is pressed. Look at the blue color cells, they run away. So this slide summarizes when WWOX deficient cell meet WWOX expression cells. There are so many factors get involved: MIF, cortectin, stress fiber, ampherin, Wnt pathway higher to tissue beta. And this accelerated migration of WWOX deficient cells, and they will undergo retrograde migration. And the wild type cell has an increased redox activity and apoptosis. And also you can see there's an increased NF-kappa B and CREAT promoter activation. So for example, in the tumor in the lung, those cells try to migrate to a lung, they must do the same way. Okay, first of all, they undergo retrograde migration and kill certain part of the normal cells, and then compromised by tissue beta before they can adhere and make lung a new home. Again, this shows a certain portion of the WWOX on the cell surface. So we try to look into which part of the WWOX can enhance cell migration, and it ends up to be in the first W domain we call 721. Repellence part is at 288, 289. So if you call the peptide on a petri dish, and the cell, when they see the peptide, they run away. Okay, they run away. But if you call 721 peptide, they migrate much faster forward, and all others are controlled. So if the peptide directly coated onto the cells, so both cells are 231, and if we caught greeting, basically no differences, they migrate forward. If we caught it one side with repellence peptide, and the other side, when they see this, they undergo retrograde migration. Alternatively, you can overexpress the SDR domain. Repellence is such a short motif, and SDR domain is huge. If you overexpress SDR domain in one side of cells, the other side of cells undergo retrograde migration. And now you can use antibody to neutralize the cell surface SDR domain so that both cells can migrate forward to each other. If you use another antibody at tyrosine 287 phosphorylation, it doesn't work. So in this slide, we summarize when cells, bacteria, or even lymphocytes, when they see greeting signal on the other side, they migrate forward. Repels the signal, they run away. Or they can adhere to it, run into it, and then get kicked out. I just show you the slide. There's an additional mechanism that is type 2 tissue beta. There's a model regarding WWOX conformation on cell surface. So cross-form we think is afforded so that 721 epitope is exposed. But once WWOX activated at tyrosine 33, in this case, epitope 286299 is exposed. So this is repellence signal. So those metastatic cancer cells in the parental tumor, they get repelled by their parent because they have dysfunctional or they do not have WWOX. So they're migrating out to seeking for a new home site. So in summary, WWOX actually has two functions. One is anti-cancer, one is post-cancer survival. So if we suppress PS14, cancer growth is going to block also neurodegeneration.